All right. So I figured I would take a step back and get kind of reacquainted with the slide that, both classes did not end in the same place. So I wanted to make sure we're all on the same uh, plane here. So just to go over where everybody did leave off, we talked about what enzymes are, the fact that they do work. They basically are the entities in which we're transferring some type of energy, either in the form of light energy or chemical energy into ATP. And the goal here, of energy is to change something. And this is what effectively enzymes do. They change their substrates in order to generate products which are chemically and or structurally distinct from the initiating substrates that entered into the active site of the enzymes. So there are two major subclasses of enzymes, and these are depending on where they exist and where they are active. For the enzymes that are released from the cell in order to then become active, so active enzymes on the outside of the cell, these are something called exoenzymes. These are typically involved in either breakdown of larger macromolecular structures, such as whole proteins, globular proteins, or complex carbohydrates, complex lipids, in order to get at the individual or single moiety of that particular macromolecule, so the cell can then activate receptors in order to import. So the concept here is that these things will have an active site specific for their substrate. Substrate breaks things down, and then the product that is then broken down can then be shuttled into the cell either by reception or simple diffusion. And these are not very common. I would say most enzymes and most cellular spaces are held within the cell, and these are called endoenzymes. So endoenzymes are enzymes that are retained in the cytoplasm, and or if you're a eukaryote, in the organelles or in the cytoplasm, right? So if you're a microbe, no organelles, so you're going to be in the cytoplasm, and the substrates have to also exist, because again, the active enzyme has to exist inside the cell, so therefore the substrate must be inside of the cell. And once again, you get a breakdown of those substrates into products. Now, as far as the reactions are concerned, the two most common kinds of reactions that you'll see in enzymology are either if you're making something, if you're pulling together chemical bonds or making chemical bonds. If you're making chemical bonds, sorry, or if you're breaking chemical bonds. So if you're making chemical bonds, by and large, especially when it comes to organic substrates and organic products, we call this condensation or dehydration synthesis. This is where we will combine hydroxyl groups in order to generate water. And since we're H2O2 and we're taking out H2O, the single oxygen that remains can serve as a bridge point between the two initiating substrates in this case, and therefore creating a chemical bond. Now, oxygen's not the only thing that can occur here, but oxygen is very typical as a merge or branch point of most molecules. Carbon is also very common. Nitrogen can be common. Anything that has multiple valencies in order to allow for the generation of extra chemical bonds. And as much as water can be used to make chemical bonds and merge two individual moieties into a singular, more complex moiety, enzymes can also exist to break them apart. And when we talk about breaking apart, we typically use water again. This is called hydrolysis. So hydrolysis is the use of water. In this case, we have water in the form of a dipole moment. It then will more or less bind to a to either oxygen or nitrogen or some other one of the other elements that have multiple valencies, and then you get the generation of extra chemical bonds. So in this case, H2, you have NH1, NH2 here, and then therefore we have an OH remaining. The hydroxide group then binds to the free carbon. Now have a loss of this carbon nitrogen bond, right? So we had a carbon nitrogen bond here. The carbon nitrogen bond is broken because of the basically nucleophilic reaction. And then we get separation because, again, once these products are formed, they no longer have the same constant of association as the substrates did. So they are unenergetically released from the enzyme. So not so surprising, even though these are the two more common types of reactions that occur based on enzymes, right? So this is synthesis and decomposition. There are other enzymes that exist that do really important biochemistry for different things. So we talked a lot about central metabolism and how central metabolism is a redox reaction, and therefore there has to be enzymes in the context of the entirety of central metabolism that can perform redox, right, or move electrons and oxidative molecules or atoms to other things. So in this case, we have different classes of redox enzymes. One of the most common, especially for microbes, is oxidoreductases. Oxidoreductases are enzymes that help break down superoxides, including hydrogen peroxide, like the production of catalase. But some of these other ones we've seen in the process of glycolysis and TCA cycle: hydrogenases, oxidases, and hydroxylases are and dehydrogenases, right? So hydrogenases and dehydrogenases are the most two common in central metabolism. And then oxidases and hydroxylases also exist as enzyme families. There are also enzymes that help move molecules from one entity to another. These are called transferases, or the class of them is called transferases. And basically, whenever you have a type of enzyme that basically attaches a functioning group or chemical group to, like, from one source to another, and then that functional group then allows for whatever it's being attached to function, or changes it changes it chemically. It's just transferring that molecule from one place to another. So one thing that we've seen multiple examples of are phosphotransferases. These are your kinases, right? Moving phosphate either from autophosphorylation or from ATP to another protein, or things like acetyltransferases and aminotransferases, or any other type of transferases typically are single replacement. There are also enzymes that do double replacement. They basically are the same general concept as transferases, but they do multi-step additions. And then finally, we have isomerases. And as we talked about, isomerases basically don't chemically change the composition of molecules, but they change their structure, or they isomerize them. So racemases and mutases are two of the more common subfamilies of isomerases in all of biology. So enzymes can be produced in a couple different ways, and they can be regulated in a couple different ways. And because enzymes perform biochemistry, and because they deplete ATP stores, because again, most of them require the function of ATP to function, we need a way to control them in a way that makes sense. And there are two different kinds of control that are typically associated with the production of enzymes, or to undergo the typical central dogma of DNA to RNA to protein. And in this case, right, we're talking about expression and function. And so the two basic expressive profiles that enzymes have, and that is most proteins, are either those that are constitutive, which means that these enzymes are produced at a constant rate. So you don't have any change in number of enzymes that you produce, irregardless of how much substrate you have. So if you have a certain amount of substrate and you produce, say, 10 enzymes per minute, if you would then go from, say, 10 pieces of substrate to 1,000 pieces of substrate, every minute you're still producing 10 enzymes, because the amount of substrate does not affect the either activity or the expression of that enzyme. And so therefore that's what we consider constitutive expression. So the ones that are just constant, they don't go up or down, they just stay at a certain level. There are also for those that, if we have one level of expression is constant expression, then the other one has to be regulated or controlled expression. And regulated control of enzymes come in two different forms. One is what we call a negative feedback loop, and that is that when you only express as much enzyme as you have for substrate, right? So if you have enough enzyme to cover substrate, and then the substrate is removed, then there's no substrate to be bound, so you express less enzyme. And this is how most enzymes and most proteins are expressed. Most proteins at some level are regulated, except for ones that are absolutely critical for cell physiology and homeostasis, also known as housekeeping genes. Similarly, the canonical wisdom is as the amount of substrate does increase, the more enzyme is expressed in order to deal with the increase in substrate, in order to, again, maintain cellular homeostasis. Because if the cell is bringing in a certain substrate or produces a certain substrate, and an enzyme is required to change that substrate into something chemically or biologically useful for a cell, you need to be able to respond to it in turn. But this helps control the amount of enzyme, because again, protein expression, protein transcription and translation are highly energetically unfavorable processes. They take a ton of energy. And then on top of that, in order to activate most of these enzymes, because again, they also require ATP, you have three different procedures just to express, translate, and then activate these enzymes. So you don't want to burn unnecessary energy by expressing more enzyme than you require. So the majority of expression of enzymes is housed at the level of transcription. So typically when we think about this process, DNA to RNA, this process accounts for about 95% of all genetic regulation of enzymes. Because again, if you're not making the RNA, you're not making the protein, and then therefore you're not getting any activity or enzymatic function or protein function, because you're not expressing the protein. However, if you spend the energy, you spend the time and the energy to transcribe the RNA and then translate it into protein, you're probably going to allow that protein to work. Now, for a lot of proteins that are expressed, there's a second level of regulation that can occur, and that is different kinds of inhibition and activation. And that is at the level of post-translational regulation. Now, there are different ways that this can exist. Now, we already talked about one, and that is the creation of apoenzymes, right? Apoenzymes are effectively, in some way, shape, or form, related to allosteric activation, in that you create an enzyme that has an inactive active site, one that's not turned on, And in the presence of a metal cofactor, a coenzyme, um, another vitamin, some other type of activator, this causes binding in it, and not in the active site, but in what we call an allosteric site, a site that's away from the active site. And this then changes the structure, because again, anything interacting with a protein is going to conformationally change the protein in order to activate and open this active site. And then when the substrate can then bind, as we talked about in class, this then allows for some other set of amino acids to move towards the active site to then perform the chemistry that's required for the change of that substrate into a metabolically active, or at least physiologically active component. Now, as you might imagine, if you can bind molecules not to the active site and in these secondary sites called allosteric sites, if we can activate proteins this way, we can also inhibit them this way. And so when you have a molecule that binds not in the active site, but because it binds to the protein somewhere else on the protein, it causes an occlusion to the active site. So therefore, the substrate is no longer recognized, and therefore it cannot be chemically modified. We call this allosteric inhibition. So why these are so important is that allosteric activation and allosteric inhibition allow for the generation of feedback loops, meaning that, as I mentioned before, a lot of enzymes do not work in a vacuum. Most of them do work in biochemical pathways. This allows for, if you have an enzyme that can modify a certain substrate and it's part of a biological cascade, it can make a product. And at some point, at the end of a biochemical process, or at the end of a biochemical pathway, there's going to be an end product. And again, because enzyme function is so energetically taxing, when you have enough of this end product, it's really important that you stop producing this product. You only want to have enough product in order to maintain cellular homeostasis. And you can always make more when you don't have enough, or if you need more, you can always get more. But when you have enough for whatever it is you need, these feedback loops allow for direct inhibition of protein function at the upstream enzymes in order to prevent the whole pathway from functioning. So in this case, these end products typically have some type of chemical binding pocket, some type of chemical interaction with most enzymes in an allosteric site. And therefore, when you have enough product, right? So when you need to make product, and if you need this much product, and then you make this much product, the excess here can then bind to the proteins that make it, and therefore cause the allosteric inhibition. So you're never going to make products directly to, you're not going to make them directly to the saturation line. You're never going to make them just to biological necessity. You'll make slightly more, and that slight more helps feed back into the system in order to prevent further activation of the protein. Now, it's also important to note that as much as we have allosteric inhibition, there's also inhibition that occurs at the actual active site. So when we have inhibition or a type of molecule, so in this case, here is an example of a substrate that can be modified by this enzyme because it has this circle domain and this triangular domain, and this is the result. But now we have this particular molecule, which has a square moiety and a triangle moiety. And now what you get is an occlusion or a binding to the active site. But because of the change in structure or the change in the chemical composition of the substrate, it cannot be chemically modified. And therefore, this molecule continuously sits in the active site, preventing or occluding the actual substrate from binding and being chemically modified. This is an example of competitive inhibition or direct competitive. Sorry. So this is competitive inhibition for the active site of these enzymes. And in either or case, allosteric inhibition or competitive inhibition both result in the same general process, and that is typically modification of the enzyme to prevent its function. Now, for what it's worth, allosteric inhibition is more reversible than competitive inhibition, simply because the constant of association in an allosteric site is not typically as high as most general competitive inhibitors, because the active site has so many different specific moieties and entities to be a binding pocket, whereas allosteric inhibition or allosteric sites are not necessarily meant to be binding pockets, but they exist as binding pockets. Now, most substrates and products most end products, typically, if they are involved in a feedback loop, again, the vast majority of feedback loops exist as negative feedback loops, because again, you want to be able to control how much of something you're producing when you already have enough. So this is an example of the tryptophan biosynthetic pathway. So tryptophan requires 3 different enzymes to go from the precursor to actual functional tryptophan. But when you make tryptophan, tryptophan actually does two really cool things. So once again, when you get to that level, so here's how much I need. Once I get to here, right, this is how much I require. When I get slightly past that, right, when I get to this point here, this excess tryptophan feeds back into these two systems, meaning that tryptophan has a binding site in enzyme 1 and prevents enzyme 1 from, therefore, making this precursor. And if this precursor is never made, enzyme 2 can't function, and the whole stream, the whole process is cut off right from the very first step. And secondly, tryptophan also binds to gene regulators. Now, we haven't really talked about transcription factors and gene regulation, but suffice to say that when you have a cell and the cell needs tryptophan, it then has proteins which know that it needs tryptophan. Remember, we talked about these internal sensor proteins. They then activate transcription factors, because again, they are factors that regulate transcription. And these then bind to the nucleoid to then produce the genes involved in tryptophan biosynthesis, or whatever it is that you need to make. And so what ends up happening is when we have enough tryptophan, when we get to this point, right, the tryptophan binds to the transcription factor and therefore blocks the ability to produce the genes that make tryptophan. And having these bidirectional switches, or these two-pronged feedback inhibitory loops, help prevent making the enzymes that are unnecessary, because you have too many enzymes for what you need for that particular byproduct or for that particular end product, and for the enzymes that still exist, because again, you've already made enough. You want to be able to inhibit them so that they do not start sucking from the pool of ATP when you have enough tryptophan in the system, or whatever the molecule is. Now, most enzymes, they exist in biochemical pathways. Different biochemical pathways come in a couple different types of cycles or pathways. One of the more common ones are linear pathways, right? So product A, enzyme 1 is intermediate B, enzyme 2, intermediate C, so on and so forth. Example is glycolysis. Most biochemical pathways exist as linear pathways. There are also cyclic pathways, Krebs cycle, the TCA cycle, right? You have multiple points of input, which means, for what it's worth, also multiple points of exput. So and you have these intermediates that can cycle around and reform the original input part that allows for a recreation of the very first step. And then you have branched. And for what it's worth, most, if not all, linear pathways have a linear pathway because they have an end product in mind. But as we've seen, even with the example here of glycolysis, different byproducts of glycolysis can come out to be part of the pentose phosphate pathway in the glycerol aldehyde 3 phosphate to glycerol to short chain fatty acid pathway. And again, all of these other examples from the TCA cycle and glycolysis for amino acid biosynthesis and nucleic acid biosynthesis. So most linear pathways are actually branched, but branched molecules or branched biochemical pathways either come in two forms. Either they're divergent, meaning that, and this is where, for example, if you were to describe how glycolysis is in relationship to the pentose phosphate pathway, this would be branched. So if I have multiple molecules of glucose, I can get multiple molecules of glucose 6-phosphate, and then I can either take glucose 6-phosphate and make fructose or 1,6-fructose bisphosphate, or I can then make xylulose and then down the ribulose pathway in the pentose phosphate pathway. There's also convergent. So this is typically when you have two different pathways that then converge. Amino acids are a great example of this because you have the amino acid group and then the R group. And typically amino acids can be made, or the amine and acid group can be made from one specific component series of biochemical structures. And then the R groups are made by a completely different set of things. And then you have them merged together in order to form your actual amino acids. So, as we mentioned before, enzymes really importantly are made in the environment that they are originally synthesized into. So all the different conditions that an enzyme is created into, including temperature, water concentration, pH, right? These are the first of three really critical components as it relates to the environment in which amino acids can fold into the different stages, right? Secondary structure, tertiary structure, quaternary and globular structure. And therefore, when you start changing temperature, water concentration, or pH, it changes the structure of the amino acid. Because again, you have amino acids which can be protonated or hydrogenated. You can have ones that can be dehydrogenated. Temperature, as you might understand, right? Like what happens when we boil pasta, and has different amino acids and different proteins in the creation of pasta or anything else that you may cook, and it changes the overall composition of those things. As far as the activity of enzymes, right? These are more function and structure, but as far as activity, right? Concentration of substrate, how much enzyme you have, are there any inhibitors, end products, which might be inhibitors or activators, and exposure to radiation, which can modify ionophores of the chemicals within a particular amino acid are all things that can affect the activity without affecting the structure, right? Or at least, I take that back. They affect the activity, and they can affect the activity. Like when you think about if you've taken biochemistry, your V max and the velocity of reaction or the concentration of saturation, um, all of these things happen in relationship to how much of substrate, how much of enzyme you have, are there inhibitors, which could be end products, or are there enough activators in order to drive enzyme function. But again, if you have activators, if you have inhibitors, these will change the structure and therefore the function and therefore the overall biochemical quantifications that you can perform on enzymes. So let's talk about a few of these things and how they are related to enzymes. And I'm kind of going to go through this a little bit quickly because we did a lot of this in the lab. So when we talk about this, again, one of the things that's really important, especially as it relates to what kind of respiration or what kind of fermentation you can perform, has to do with the availability and the toxicity of oxygen. And the availability or toxicity of oxygen really helps define how certain enzymes can work because of their ability to either oxidize or add oxygen, or if they're a transferase, can function in that particular manner. And so what we get is a series of different types of growth in different concentrations of oxygen, which lead to different classifications of microorganisms. So the first are obligate aerobes. Obligate aerobes are obliged, or they're obligated to live in an aerobic environment, meaning that they have to perform aerobic respiration. They cannot be fermenters. They are typically fast growing and need to live in aerobic environments. Examples that are really kind of easy to spot: Mycobacterium tuberculosis and Pseudomonas aeruginosa, two distinct bacteria that infect the lungs of higher order mammals. And why would you infect the lungs? Because it's highly, highly oxygenated. We then have microaerophiles. Microaerophiles are they are aerobic and perform aerobic respirations, but they need to live in a certain percentage of atmospheric oxygen. Again, as we talked about in class, oxygen makes up about 20% of atmospheric composition of gas, and they need to live in oxygen environments which are 2 to 10%. So there's got to be some oxygen, but not a ton. And one really good example of this is Helicobacter pylori. Helicobacter pylori is a pathogenic microorganism that is typically commensal in most people, but it lives inside the stomach. So the stomach is not extremely aerated, but it does have some aeration because of the function of stomach acid and churning and the need for oxygen to exist to produce mucus inside of the stomach lining. We then have facultative anaerobes. These are really kind of the happy medium of all these types of cells. They will grow aerobically until oxygen is depleted, and then they can continue to grow extremely quickly because they are very adept at performing anaerobic respiration, meaning that they can respire without using oxygen as a terminal electron acceptor, or they can, in some cases, they can even reduce other elements that are not carbon-based in order to perform respiration and transfer of chemical energy. Two really common examples of this: Escherichia coli and Salmonella enterica. We then have aerotolerant anaerobes. So these are anaerobes that would prefer to grow anaerobically, but they can survive and do okay with oxygen being very fluctuating. Streptococcus genus is very good at this. So ones that live in the back of your throat or in your lungs, but on the more the outside of the lung rather than the inside of the lung. So Strep pneumoniae lives inside the lung, and Strep pyogenes inside the back of the throat. And then finally, we get to these guys here, obligate anaerobes. They typically are either fermenters, or if they have found a way to have an electron transport chain and a terminal electron acceptor by differential redox reactions of inorganic molecules, they can perform anaerobic respiration. One of the most common genus that you'll see, especially as it relates to human health and disease, are the Clostridia, both the Clostridia that exist that make really important immune modulating molecules like butyrate and are involved in the gastrointestinal tract colonization, as well as Clostridium perfringens, which is the causative agent of gas gangrene, and Clostridium botulinum, which is the causative agent of botulism toxicity. Temperature also plays an important role in defining certain aspects of bacteria. There are bacteria whose growth rate, when their growth rate is optimally in different temperature ranges, gives rise to a certain classification. So we have psychrophiles, which grow at extremely cold temperature, mesophiles, which grow mostly in our biosphere, so between 15 degrees centigrade and 45 degrees centigrade, which, just for reference, 37 degrees Celsius is equal to 98.6 degrees Fahrenheit. So you're talking about maybe 115, 120. So that's really at the kind of the end of where we're really, can live and our bodies can handle that kind of heat. Anything above that, we get into the thermophiles. And for those that live in like the deep sea thermal vents in volcanoes and volcanic ash, we have the hyperthermophiles, which live up to degrees close to 110 or 115 degrees Celsius. pH, again, the availability of H plus protons, not only important for ATP synthesis and the enzymes and the transporters that are involved in ATP synthesis, but also because any amino acid that is negatively charged can be protonated, meaning that the more H plus there is, the more protonation that can occur, which affects the amino acid structure, which then therefore affects the protein structure, right? So if protein structure is highly related to amino acid sequence and composition, it also means that amino acid sequence and composition, the structure of each individual amino acid, plays an important role in denoting the structure of the protein. So being able to withstand free hydrogen protons really gives rise to the ability to grow in certain places. And again, this has to do with growth rate, not the ability to survive and be static in a particular pH. And we can get to what I mean about that in a minute. But this is where these bacteria are happy to grow. So if you have microorganisms that grow between pHs of 0 and 4.5 or 5. You have a bacteria that's called an acidophile. A really good example of this is Helicobacter pylori, which grows inside the stomach. So H. pylori grows inside of the stomach, and its highest growth rate is in that particular pH. So it's happy to grow in pHs of 2, 1.5, in around stomach acid. We then have neutrophils. They grow in pHs close to what our pH norm is, between 5.5, 6, all the way to 8.5 or so. These are your E. coli, Salmonella, Streptococcus, Staphylococcus, most, if not all, human pathogens. Sorry, not most, because Helicobacter can be a human pathogen. So most human pathogens and most human microbiota, and microbiota that live in most animals and plants. And neutrophiles, then alkylophiles are those that live in pretty contaminated environments that have high levels of alkalinity. So places where there's high levels of hydroxide. So former bleach plants, or when bleach gets into waterways, or certain batteries that give off the second half of the salt bridge, so not battery acid, but alkaline aspects of batteries, is where you'd find most of these particular microorganisms. And for what it's worth, most nutrophiles, if they are human or animal pathogens, or human or animal microorganisms, commensal microorganisms, they do have some capacity to survive in stomach acid in order to then get away from the stomach acid. But that doesn't mean that they grow there. It means that they can survive it. So, for example, E. coli, Salmonella, Clostridium perfringens, some of these other human pathogens that infect the gastrointestinal tract, you eat them, so they pass through the stomach, but then they infect the intestines and not the stomach. We also have osmotic regulation. This has to do with how much salt you can withstand versus how much water. There are halophiles, ones that love to be in a high concentration of salt, and then osmophiles, which love to be in high levels of water. So halophiles you might find in a place like the Dead Sea, and osmophiles would be would be microorganisms that would live inside of waterways, so fresh water, ponds, lakes, streams, etc. So it's important to remember, right? There are two really important critical aspects to physiology and metabolism. They are the ability to survive, and then the ability to survive in order for the function of replication. And in order to replicate, it takes a lot of different enzymes to not only build the other cellular components to make a daughter cell, but also to be able to divide the most critical of all the features that need to be divided amongst cells that are dividing, and that's the DNA. So here's kind of a schematic of DNA replication. We're going to cover this in genetics, so I'm not going to spend a ton of time on this, but this is just to show you that there's a lot of, when I tell you that this is an energetically difficult process, I mean that because look how many enzymes we have here. You have helicases, topoisomerases, gyrases, DNA ligases, polymerases, and of course the ability to make or find free nucleotides in order to keep generating new strands of DNA for replication. The same thing is true for RNA transcription. We have polymerases, gyrases, helicases, topoisomerases, and we need more free nucleotides in order to make the mRNA transcript that we're making. Now, the last couple slides here have to do with so we talked a lot about metabolism and the usage of transferring energy from one source to another, but we haven't really talked about how organisms capture nutrients. And in order to understand how these microorganisms live in an environment, we first have to understand that what is the general condition in which these microorganisms do live in the environment. And there are many manuscripts out there that would suggest that the vast majority of microorganisms, well over 95, 96, 98% of them, live in something called a biofilm. And biofilms are these cyclic, evolving colonies or communities of microorganisms that exist and have their own zip codes and own environments, and they play a really critical role in the survival and metabolism and the ability to signal between different members of the community. Now, all biofilms have a very similar pathway in that they involve free-swimming bacteria that then find a place of attachment. And then once they find their place of attachment, I want you to appreciate this, they have flagella here but then they stop producing flagella once they have started to create their slime layer. So when they then engage in irreversible attachment and proliferation, they start to merge their capsules and start to merge their slime layers in order to form what they call a glycocalyx. When that glycocalyx continues to grow because the number of individuals continues to divide, this is called biofilm maturation. And then over time, just like any other environment, including microenvironments, um, these environments only have a certain carrying capacity, which means that these cells then need to, when cells start to die, because there's not enough nutrients to go around in that particular environment, some cells will break open the biofilm, re-establish their flagellar expression and their key system in order to swim away, in order to find a new site of attachment in a new niche space. All biofilms have a very important gradient in that the cells on the outside are far healthier than the cells on the inside. And so the cells on the inside and the amount of cell death that occurs on the inside is really, really important in order to gauge the relative structure and health of the biofilm. Because as nutrients pass by these biofilms, right, they are going to be taken up by the cells closest to the edge. And same thing with oxygen or other dissolvable gases like carbon dioxide, etc. They are going to have the most access to these things. How now the interesting thing is that, again, because these cells on the inside are maintaining the health or the relative health of the biofilm, because again, the more nutrients, the more oxygen or other gases that make it into the biofilm, the more of these things are going to get to the cortex or the inner part of the biofilm, meaning that these cells in the middle are really important for generating most of the signals that are involved in maintaining harmonic genetic responses, also known as quorum sensing, which can help change physiology, expression of virulence factors, expression of physiological factors, the expression of genes involved in generating certain substrates and byproducts, et cetera, so on and so forth. And because these cells are growing at such a rapid rate, especially on the outside and more slowly on the inside, there's far more genetic diversity on the outside and fewer genetic diversity or lower genetic diversity on the inside. And this is really important because when you have more genetic diversity on the outside, it means that you have greater chance to have cells that exist on the outside that can be really important for the generation of things like antimicrobial resistance and activation of or acquisition of genes involved in nutrient acquisition that you might not have had before. And when these strains exist on the outside of the biofilm, when antibiotics then hit the first layer, they're repelled out of the biofilm by these resistant bacteria. And of course, like I said before, there's a lot of cooperation because of the cell signaling, and there's constant competition because, again, the health of the biofilm depends on fitness. So you want the most fit bacteria to exist where there's most amount of nutrients, and those then, therefore, in order to maintain the regular structure of the biofilm, are really important for retaining enzyme, absorption of those resources in order to generate byproducts to make sure that all the cells on the inner cortex remain alive and healthy. So I'm just going to cut it here because the rest of this kind of goes over everything that we talked about. It's important to understand that a lot of these systems are really critical for bacteria to survive and thrive. One last thing that we'll talk about real quick is that most of the communication within a particular biofilm happens to be through quorum sensing, and obviously, so the cells that have the highest density of cells are going to give off the highest number of chemical interactions. But these chemical interactions do have to play an important role in regulation of gene and harmonic genetic response. as it relates to how other bacteria are connectedly interacting with one another. So there are things like nanowires, cell-to-cell contact, and other types of interactions, which really play an important role in modulating how much of those chemical signals can play on regulating gene expression. So that's all of that, and I hope you guys enjoy.